Good morning, bio. I know you guys are so sad that I'm invisible again and I'm a ghost. It's okay. I still love you guys. All right, here's your warm up. You guys know what to do. Bring out your notebook, write all these things down. You have about 15 minutes, and after 15 minutes, your sub will continue this video. Go. All right, good job. I will check all that later. You are welcome to leave your bio notebooks in the classroom if you want, but please make sure that you have whatever you're going to need for your midterm, since that's going to be due. Your midterm project's going to be due pretty soon after we get back from Christmas break. So if you need your bio notes to do that, then you should probably take them home. All right, you guys remember this. This was your pub quiz. Because I'm so amazing and you guys know I'm so amazing, there were some issues with this, and I think generally people did okay. So what I've decided to do with your grades, if you go on to facts, is you will see that your grade on this pub quiz is extra credit. So to those of you that got really amazing grades, your grades just bumped up by a lot. And if you didn't think you did so great, that's okay. It still bumped up your grade as well. And I know there were some issues with a few of these. They're kind of hard to tell. And again, the lesson is that when you're in a real life laboratory, it's not so cut and dry. You don't automatically know, like, this is obviously metaphase, okay? Obviously. This is obviously anaphase. So is this one. This is interphase, this is interphase, this is interphase. Those ones are obvious. But yeah, like this one, not so much. And I actually gave you guys a bonus point for that one. And this one, it's like, is it anaphase or is it not? Well, it's not, so this is prophase. So it's kind of hard to tell, okay? But anyways, that's all extra credit. So please review those steps, because today we're reviewing mitosis and meiosis. Let's go over some key terms again. The good thing is that if I'm speaking too fast, just ask to pause the video and take your notes. Diploid. That means that we have two sets of chromosomes. Okay? This ploid, P-L-O-I-D, means a set of chromosomes. Di or di, right, means two, right? Diploid means two sets of chromosomes. You have one set from mom and one set from dad, from each biological parent. Okay? And well, never mind. All right, meiosis. We know what meiosis is. We just barely talked about it. But it is the creation of sex cells. So we have interphase, which starts off with the diploid cell, and then we don't go through interphase again. Okay, we go through interphase one time, and then we start meiosis 1, which is PMAT, right? P-M-A-T, and each of those have a 1. So for example, prophase 1, metaphase 1, anaphase 1, telophase 1, and then we go on to meiosis 2, no interphase again, no interphase. So meiosis 1, P1, M1, A1, T1, and then go straight to 2, P2, M2, A2, T2. So let me just say the whole thing over again, okay? We are going to go through interphase, prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis, prophase 2, metaphase 2, anaphase 2, telophase 2, cytokinesis. Meiosis is a reduction division, if you remember that term, reduction division, that results in 4 daughter cells, and these 4 daughter cells are half the number of your diploid cells, so these cells are haploid, right? Haploid, meaning one set of chromosomes. And so, see how we have a chromosome here? Well, we have half the set of chromosomes here. Don't forget that the second meiosis 2, the second set of meiosis here, the second, division here, is basically the exact same thing as mitosis. It looks exactly the same, where we end up with chromatids as our daughter cells, but meiosis 1 is where we have differences, okay? So when does crossing over happen? Well, that happens during prophase 1, and yes, you'll have to be able to know the 1 versus the 2, okay? We talked about this. right? We know that meiosis creates gametes. Gametes, so in males, that means that we're creating a sperm, and that in females, we're creating eggs. So from one diploid, we end up with 4 sperm or with 4 eggs, respectively. And they contain half of the genetic material. That makes sense because in order for us to have a diploid or an organism that has the correct number of chromosomes, then mom needs to have half and dad needs to have half in order for offspring to have a complete set. Okay, and when we're talking about the original cell, like this one during meiosis, like what's this cell? Where does that come from? That is from the spermatozoon and from the ovum. They are the ones that are diploid, and then they go off to divide and create the actual gametes. Crossing over, we already mentioned, happens during prophase 1 of meiosis 1. And that's the exchange between these homologous pairs. So here's a homologous pair. We're going with blue for dad, red for mom. And then they stand next to each other. Remember how Richard and I switched hands and legs and a pinky and stuff? Right? So here they're literally switching out because remember, this is an actual physical thing inside of these cells. So because they're a physical thing, they can actually swap legs and connect at just the right spot. Okay, so because maybe mom has blue eyes, dad has brown eyes, and then they switch. So this specific chromosome now codes for mom's blue eyes, even though the rest looks like dad. And here, this mom looks like it has dad's brown eyes because they crossed over. This is a actual physical combination that's happening, as if Richard and I were actually popping out our legs and replacing them onto each other. Like, I mean, it's not so crude, but that's basically what happens. Okay. Independent assortment. We briefly touched on this. You should probably know this term. Is when different genes independently separate from one another. Okay, so for example, here is this parent cell, right? We have dad's parent cells, mom's parent cells. Well, when we separate them through meiosis, we have a whole slew of combinations that are possible, right? We could get this side, see how this chromatid is this light blue chromatid with this dark blue chromatid and this dark blue chromatid, right? But it doesn't necessarily have to happen that way, right? We can get this one, we can get this one. And so because there's so many possibilities, and don't forget crossing over has happened, and that you're making, well, I looked it up just so I could be sure, and it's just under 200 million sperm per ejaculation. And plus, with the multitude of eggs that are already present in a female, since those are created while the female is in utero inside of her mother's womb, the actual possible combinations of which egg cell happens to come out during ovulation and which sperm happens to reach the egg first and appropriately combine the genetic material inside the sperm with the egg creates the fertilized egg, which creates a new person or a new being. If it's a dog or a turtle or a rat or something, right? They all do the exact same thing in terms of sperm and egg and how independent assortment can create a multitude of varieties, right? And those answers should answer your warm-up question, because that is why siblings can look vastly different from each other, yet still look somewhat related, right? Because they come from mom and dad. And here's just a review for haploid, which we talked about. That's a single set of unpaired chromosomes, so gametes. So which cells are going to be haploid? Out of the whole entire body, which cells? The sex cells are going to be haploid, okay? An eye cell is diploid. The tip of your finger is made of diploid cells. Your nose is made up of diploid cells. Your pancreas is made up of diploid cells. However, the gametes are haploid. And we also have segregation, okay? This means just a separation to not be near each other, okay? We're talking about bio, but the root word is the same as in, history. Okay, so that's when you have paired alleles, right? We know this word already, alleles. You should be familiar with this word. Paired alleles, they separate when gametes are being formed, in other words, during mitosis, so that each gamete contains one copy of every paired unit. For example, right, we talked about trisomy 21, right, which is Down syndrome. That did not properly segregate its alleles because now we have multiple copies of chromosome 21, right? Normally, we need to make sure that each one has its own pair of alleles. And we talked about this. See, we even have, like, let's say we have a heterozygous parent, we separate the alleles, and here are the gametes. So then we can decide what you're going to have, right? This parent, this, let's say this is a sperm. This sperm happens to be dominant for a trait. This one happens to be recessive for a trait. That would make sense because if a parent is heterozygous, then they could either donate this A, this big A, or this little a, as we saw with our Punnett squares. And so we would only get one, and mom in the egg would donate whichever one she happens to be, if she's heterozygous or homozygous dominant, homozygous recessive, etc. Okay, we know these terms. If you do not know these terms, then I highly suggest that you look at the stuff that we talked about before. Okay, and that is all online on the other slideshow under Chapter 5 of Google Classroom. And finally, heredity, which we've talked about throughout Chapter 5, is when you pass down the characteristics genetically from one generation to another. Okay, I know that we're using the term mom and dad loosely here. What I'm talking about is biological father and biological mother. And so here's mom, right? She's got mouth open, teeth and tongue sticking out. Here's child, has the same thing. But child did not inherit her nose, and child inherited biological father's nose. Eyes, which one does he look more like? I would say he looks more like dad. Eyebrows, I'd say probably more like mom's, right? Hair inherited from father, right? So here's all these little differences, right? To make someone that looks completely different and who is an actual unique individual separate from his or her parents. All right, so what you guys are going to do right now is before you watch this video, what I want you guys to do is go through, make sure that you understand all these terms. If you need to get on your Chromebook and make sure that any of the terms that I said that were not necessarily on here, like if you don't remember what a Punnett square is, or you don't remember the term heterozygous dominant and things like that, I highly suggest that you go through that. And once you are done with that, you will watch this video. It will be up to the discretion of your substitute teacher if you will play this on the big screen, or if you guys will be watching this individually on your Chromebooks or with pairs. I honestly, it's however you guys are behaving, and it's up to your sub. They have my full being, word. Again, I'm not teaching English at the moment, okay? They can choose what they want you guys to do to compare. Don't forget that you can choose mitosis or meiosis for your midterm project. You can also choose photosynthesis, cellular respiration, or Mendelian and non-Mendelian genetics and inheritance. Okay, so you're going to choose one of those for your midterm. I will give you guys time after Christmas break to work on that. I would highly suggest that if you have some extra time during class today, that you get that done. I know it's almost Christmas break and Mrs. Prosser is still teaching us stuff and she's not even here. This is so not fair. Yeah, okay. Remember, science waits for no man. So please, I highly suggest you take advantage of this time and do that. Or make sure that your lab notebook looks really, really, really well looks really good, because it's going to be collected January 10th. Your midterm is due to me January 12th. That's not that much time, considering that we're also going to have a field trip. Okay, if you need to turn in your permission slips, please hand them to Mr. Ganter. Okay, hand them to Mr. Ganter, please. Or you may also have your parents email me a signed copy of your permission slip. You guys are amazing. Go through these terms, finish everything up during Christmas or before Christmas break. I know you guys can do it. And I want to see beautiful, beautiful children's books. I'm so excited to read them. I can't wait. You guys are amazing. Okay, I love you guys. You guys are wonderful human beings who do amazing things and who are capable of so much. Have a wonderful Christmas break. I'm so sorry I couldn't be there to say bye to you guys, but you guys are seriously so amazing. Okay, bye.